on Blackboard, I'm nervous. Here we go. Okay, so on Blackboard, I have a folder that says videos in today's folder. And the actual videos are there where you could like download them to your computer, but sometimes people that don't have the proper players or whatever to play the different ones. So in addition, I've added most of these are on YouTube now. Everything's on YouTube now. So I've just added the YouTube links as well, and they should be clickable links. I just clicked on this one earlier and set it up so that I didn't have to have the ad. I could skip it. So now you know how to get to it too, because I'm going to play it for the class. I'm going to take my headphones off, but I'm not sure how well it's picking up the audio when I actually play them. So what we shall see.
Leukocytes are white blood cells that help fight infection. At sites of injury, infection, or inflammation, cytokines are released and stimulate endothelial cells that line adjacent blood vessels. The endothelial cells then express surface proteins called selectins. Selectins bind to carbohydrates displayed on the membrane of the leukocytes, causing them to stick to the walls of the blood vessels. This binding interaction is of sufficiently low affinity that the leukocytes can literally roll along the vessel walls in search for points to exit the vessel. There, they adhere tightly and squeeze between endothelial cells without disrupting the vessel walls, then crawl out of the blood vessel into the adjacent connective tissue. Here, leukocyte rolling is observed directly in an anesthetized mouse. The up and down movement of the frame is due to the mouse's breathing. 2 blood vessels are shown. The upper one is an artery with blood flowing from right to left. The lower one is a vein with blood flowing from left to right. Leukocytes only adhere to the surface of veins. They do not crawl out of arteries. Some leukocytes are firmly attached and are in the process of crawling through the vessel walls, whereas others have already left the vessel and are seen in the surrounding connective tissue. When the blood flow is stopped temporarily by gently clamping the vessels, we can appreciate how densely both vessels are filled with red blood cells. Red blood cells do not interact with the vessel walls and move so fast under normal flow that we cannot see them. When the blood flow is restored, some of the leukocytes continue rolling, whereas all non-interacting cells are immediately washed away by the shear.
What else?
I definitely don't want this next one to play, whatever it happens to be. Close that out. Okay, so that's definitely one of my favorite ones, and I actually wanted to rewind. I was so worried about the other one playing. So now I have to endure that ad. Okay, I'm gonna hit pause. Right, there we go. Okay, so what I like is you actually get to see the real thing happening. How they did that, I have no earthly idea, but it's really super cool. Definitely some of the coolest stuff. So I started out with inflammation because this is paramount to your immune response, right? These white blood cells getting to the area where they are needed to fight infection or even to clean up damaged cells, injured cells, right? Even when we don't have invasion happening. So as you can see, there are multi-steps in the process for this to happen, and we're going to go through each one of the steps in detail. So this is one of my favorite videos. Another one that I have is the homing one, and hopefully we don't get an ad for this one.
Didn't look
There we go.
visualize lymphocyte homing to a site of injury, a zebrafish larva was anesthetized and its fin pierced with a needle to introduce a small wound. A vein is seen at the bottom of the frame. Because the fin is very thin and transparent, we can watch directly as lymphocytes crawl out of the blood vessel and migrate towards the wound. They are attracted there by chemicals released from damaged cells, invading bacteria, and other lymphocytes. In a zoomed out view, we can appreciate that lymphocyte invasion is restricted to the wounded area. The static cells that are dispersed in the connective tissue are fibroblasts. In these movies, 60 minutes of real time are compressed into 15 seconds.
So that one is super fast, right? So they even tell you they sped that up. You can definitely tell they're really moving really fast. But to give you appreciation too, that they're called to a particular area, right? This is only going to happen when they're signaled to go to that particular area. So let's see how many of my anatomy and physiology people are here. See if they know the answer to this one. So they talk about how one's an artery and one's a vein. And they said it only crawls out of veins. Can anyone give me some reasons anatomy-wise they only crawl out of veins instead of arteries?
Pressure's not as high.
Pressure's not as high in your venous system.
It's a reservoir, the veins are a reservoir.
The veins are kind of a reservoir. The blood tends to somewhat pool there, collect there until it's pushed along.
It's not a danger to the body for a cell to squeeze through the wall of a vein as much as it would be for an artery because the higher pressure could
Right, the higher pressure, it could create problems. What did someone else say?
Smooth muscle.
Right, there's a smooth muscle associated with your arteries, where with your veins there's not as many layers, right? And we're talking about little small venules in your capillaries more so where this is happening. So, maybe 1 cell layer thick vessels, right, that these white blood cells are crawling out of, okay? Under a lot less pressure, less material to move through, just 1 cell layer usually. So we're talking usually about capillaries, very small venules. So not under as much pressure. And as I said, this interaction happens just for the white blood cells because as you saw, just for the white blood cells do they actually have the selectins, right? What we'll call adhesion molecules later, right? Where they're actually able to interact, grab onto, and again, only these selectin molecules are expressed when there's that signaling. So notice these little dots are representing that signaling. It's all chemical messages that are being sent, right? So we'll talk about specific chemical messages that are sent that'll elicit this response that we call inflammation.
So we need to know that it binds to a carbohydrate?
No. Carol asked, do we need to know if it binds to a carbohydrate? No. We don't need to know it. And some of these videos and stuff, they're going to go into a little bit more detail sometimes than I am. I'm definitely going to review with you guys the level that I want you guys to know. Okay, so that brings us to the PowerPoint for today. So you'll notice that we're going to outline this process of inflammation, right? What are the steps in the process? We're really going to focus on the factors that initiate this process, right? So things like tissue damage, invasion, release of those chemical signals, right, that are going to call the cells there. What is the benefit, right? What's the outcome of inflammation? Why do we want those white blood cells to get there? Anyone have a clue or a guess?
To keep us from getting sick.
Right, those white blood cells, some of them can do what to those invaders? They can eat them, phagocytize them, kill them, right? Some of them can release chemicals. Others are going to elicit part of our adaptive immune system that we're going to talk about later that will produce antibodies to help us gather up and destroy even more of the pathogens that get in. So I'm referring to right here, I'm referring to those 2 videos we just watched, right? The leukocyte rolling and leukocyte homing. They're in that video folder for today's lecture with also YouTube links to make it easier so you don't have to worry about downloading the actual videos that I have. So in this PowerPoint is the textbooks animation, and as I said, I'm going to add a whole button for you guys, and it's going to be, I just went through on the internet and found the links, and so it's in some other textbooks, but it's all the same publisher, McGraw-Hill, that have all these animations for, for immunity. And some of them will even be in your Connect homeworks. But again, I just wanted a quick reference for you. And so the other thing is that some of them have little quizzes associated with them, and they say you can email your instructor. Do not email me. Okay, I've already gotten one from my other class. I was like, someone must not have listened to me, right? But you can take the quiz, it'll give you your results. Hey, it's even more, interaction, right? Help you guys learn this material. So I'm going to go ahead and play this one, and then we're going to discuss the different attributes. And so you'll see a little bit different details in this one. They're going to mention some things that weren't mentioned in the other video.
The inflammatory response is an important, nonspecific defense against tissue damage. It begins when injured tissue cells release chemical signals that activate the endothelial cells of nearby capillaries. Within the capillaries, adhesion molecules called selectins are displayed on the activated endothelial cells. These adhesion molecules attract neutrophils, slow them down, and cause the neutrophils to roll along the endothelium. As the neutrophils roll along the endothelium, they encounter chemicals that activate integrins, which are adhesion receptors on their surfaces. These integrins then tightly attach to adhesion receptor molecules on the endothelial cells. This causes the neutrophils to stick to the endothelium and stop rolling. This accumulation of neutrophils along the walls of the capillary is referred to as margination. The inflammatory mediators released by the injured tissue bring about changes in the environment that cause mast cells to degranulate and release histamine. Histamine causes vasodilation and an opening of the junctions between the endothelial cells, allowing fluid and leukocytes to leave the capillary and enter the infected tissue. The neutrophils now undergo dramatic changes in shape and squeeze through the endothelial wall into the interstitial tissue fluid. This process is called extravasation. The neutrophils, followed by other types of phagocytes, are attracted to the damaged site by chemotactic substances released by bacteria and tissue breakdown products. They ingest and destroy invading bacteria.
So as I said, this one goes into a little bit more detail. So they're naming a specific type of white blood cell that we'll discuss later called neutrophils, right? And as you can see too in the animation, they're able to do phagocytosis. They're able to gobble up, right, and destroy the invaders directly. Mast cells are also another type of white blood cells that are present in your tissues. And these are a type of cells that have what we call granules inside them. They're vesicles that contain certain substances. And so these particular cells are similar to another set we'll talk about, which are basophils, which contain the granules that contain histamine. And as I said, histamine is a very good vasodilator. It's going to help facilitate the movement of these white blood cells into the area. And that's the whole main reason for this inflammation, right? This movement of white blood cells to the area to destroy the invaders, to trap them, right? To keep them from spreading throughout the body. Now, so we'll look at specific white blood cells and specific roles as we continue our discussion through immunology. There was something specific I wanted to say, and it right out of my head. All right, it'll come to me. Oh, I know. Okay, so leakage of fluid. So we all, at one time or another, have seen or experienced inflammation, right? One of the things, what are the cardinal signs? What are things that you can see when you have inflammation?
Swelling.
Redness, swelling, you can feel heat. Sometimes you experience pain, right? And a lot of this is attributed to what they say is going on here, okay? So the fluid attributes to the swelling, right? So that leakage of the fluid. And that's just a byproduct of the white blood cells leaving, right? That vasodilation, the increase in the diameter of the blood vessels to allow. And again, in addition to that fluid leaking out, how did those white blood cells get there? The blood is traveling there, right? And in your capillary beds, you don't always have flow to particular areas, right? It can be closed off and different amounts can come in at different times as need is expressed by the tissue, okay? So in this case, they're definitely saying we need blood because we need those white blood cells. So you get that increased blood flow. That increased blood flow is the redness, right? More blood to that area. That capillary bed is just full of blood. Make sense? Okay. The heat is also due to your blood, right? Your blood is the warmest component of your body. It carries most of the heat, right? And it also helps you cool off, right? When you get in a heated situation, when you're exercising, you're producing a lot of heat. This is why at first your skin will get really red, because your skin is the largest surface area for that heat exchange to happen, to get rid of that heat. So again, the blood flow increases to your skin, not necessarily because your skin is working as you're exercising, but it's working when you're exercising to get rid of that heat, right? So if you increase blood flow to the skin to get rid of the heat, right, then you start sweating, then you get the convection, and you get even more cooling that happens. So the redness is the increased blood flow, right? So is the heat, okay? So probably the easiest way to remember too and to think about that exchange is elephants are the best example of this. Which do we have at the zoo? Anyone know if we have Asian or African elephants? We have the Asian. Anyone know a really quick, easy way to tell?
Size of their ears.
The size of their ears. So the Asian ones are smaller, right, than the African ones, right? If we had African ones, they would have those big, huge ears. And the reason for that is the African ones live in a much warmer climate than the Asian ones. And they literally pump their blood through their ears, and that's why they're fanning their ears, is to cool themselves down, to cool their blood, right? So they have those big, huge ears to help with that, and that's why they're so thin, right? To help with that cooling mechanism. We have our skin, right? We use our skin for that. So we're going to get rid of that heat, right? And that's why that increased blood flow is associated always with our blood, and it's why you have the increased heat. The blood flow is the redness. The swelling, right, is the leaking of that fluid. Now the pain has to do also with the swelling, right? That fluid then stretches the area, pushes up against pain receptors, right? So now we know why we get red, warm, right, and swollen, and why it hurts. But there is a reason behind why this happens, right? So as they said, there are some cells, resident cells in the area. So there's a certain set of phagocytic cells we refer to as macrophages. So you'll see this one here, he's right here. As that punctures through the skin, they're on site. They're going to start gobbling up. They are going to release chemical signals too, right, that are going to cause the changes, call up the blood vessels, make them dilate, get the more of the cells to come, mostly neutrophils because they're our main phagocytic cells, right, and more lymphocytes, specifically, not lymphocyte, why did I say lymphocyte? Monocytes. Monocytes is the name given to your macrophages when they're in your blood. When they travel into your tissues, they differentiate into the cell type we refer to as macrophages. And we're going to go over this again later, right? So don't stress about it. But the more I say it, the more likely you're going to remember it, right? And so that's why here in this picture they say a recruited macrophage, right? So this is one that traveled to the area. He didn't live here. This wasn't his home. He came and joined the fight. And then the neutrophils, these guys really only travel into your tissues during inflammation, right? They're going to be called there during inflammation. And they're really cool cells. They're very powerful killing cells. And I nickname the suicide bombers because they'll even kill themselves in an effort to and destroy the invaders, right? They are serious about killing. So The purpose is contain the site of damage, right? Localize the response, as we saw with that homing video, right? They're just going to the area where they're being called. We want to eliminate the invader, right? Gobble it up, destroy it. And we want to restore tissue function. And that's the fifth sign that sometimes you get with inflammation is loss of function, right? And that unfortunately sometimes can be bad, depending on where the inflammation is. So how is it that those cells know that there's something bad going on? They have receptors on their surface, and they use lots of acronyms, right? I hate them, right? But really the important thing is that we're looking at specific patterns, right, that we're recognizing with these receptors. So some examples of ones that we'll talk about are toll-like receptors, which is what the TLR stands for, NOD receptors. These just stand for, and sometimes I even forget, so pattern-associated molecular, or pathogen-associated molecular patterns, right? So pathogen, right, is something foreign, something bad. Danger, right? So these are all things, again, we're looking at particular patterns, right? And this is part of our innate system. This is what you're born with. So your body already kind of knows a general idea, has receptors for the general aspects of bacteria and the bad guys, right, that we commonly see. So those host cells, right, that get damaged or these receptors get triggered, they're going to release cytokines. These are chemical messages. An example of one is histamine, right? So this is how they communicate. These are their voices, you could say. They're going to release these chemicals, as you saw. They're represented different ways in the different animations. Right? And these are going to bind to receptors on endothelial cells, right, on the phagocytic cells, and call them to that area. And then the microbes themselves, right, they release substances not on purpose, just because they do, right? They have byproducts and stuff that they produce. And our body is able to sense those as being foreign and be attracted to them, right? So the microbes themselves, right, is, you almost could say we can smell them, right? We can smell them, we chase after them, right? We've got their scent down. So if the blood vessels are damaged, two enzymatic cascades are activated that lead to the clotting, because we don't want the blood uncontrollably leaking into the tissue. We also don't want organisms getting into our blood, right, and then using that superhighway to get from one area of the body completely to the other. I mean, sitting here at resting heart rate, you can literally move within a minute from one side of the body to the other. Like, you could make a complete circuit and be back. That's how fast your blood is traveling. So, as we said, that dilation of the blood vessels, that is with that greater blood flow, is the redness and the heat, right? Your blood is the redness and the heat that you see. Although it's going to run at a relatively slower rate, but we're going to increase the amount going to that area, right? It's not always completely open. And then the leakage of the fluids leads to the swelling, and then pushing up against the pain receptors, and we feel that pain. So margination, right, that movement of the leukocytes, another name, white blood cells, right, that refers to all of them. And as we saw, those endothelial cells, they produce those selectins, right, those molecules on their surface that allow the phagocytes to grab on. And I think our fast-talking guy talked about diapedesis, how it meant oozing, right? So this is that oozing process. Clotting factors are going to wall off the site of infection, right? And we're not going to go through the clotting cascade in this class. Yay. Dead neutrophils, because remember I said I called these guys suicide bombers, right? They will kill themselves sometimes in the process of trying to eliminate the invaders. So when you experience pus, a lot of times, that oozing stuff is your dead cells, mostly your neutrophils. But the reason why you want to avoid pus is because it can be contagious, because it also would contain the pathogens as well, right?
Even though hopefully the pathogens are dead, it's just the little particles that are in.
Yeah, hopefully, but there's no guarantee, depending on what's going on,
Yeah.
right?
And
The
like
level
that same
infection.
volcano,
Yeah.
sort of boil thing.
Yeah. We'll talk about that later. Okay, so there's two different types of inflammation. Acute inflammation is short term. It acts really fast, and if we are able to clean everything up, then it's going to subside, right? Acute means short acting, right? It's going to fast acting, short duration is the quickest definition of acute. Chronic is when acute fails, right? When acute can't get the job done, right? That very first rush in, we're not able to maintain it. And so then you get long-term inflammation. And this can be bad. This happens in a lot of disease conditions. So probably the best example of this is Mycobacteria tuberculosis that causes tuberculosis. So when this gets into the lungs, the resident macrophages, the alveolar macrophages can engulf it. But as we'll talk about later, we aren't always successful at killing the microorganism. And this particular one can grow inside your macrophages, right? So they're not able to kill it. They're signaling to the other macrophages that are coming in with the inflammation, and they're just going to keep piling up on this guy and engulfing, and it's just going to keep growing and growing inside these macrophages. And then even T cells sometimes will get involved, and you get this mass of fused macrophages called giant cells, and then with the T cells you get these granuloma formations. Now the bad news with this happening in your lungs is that then you have decreased area for gas exchange, right? So eventually it could kill you, which before we had antibiotics to treat tuberculosis, that's what eventually happened, is they slowly died, right? From slowly losing the ability to be able to breathe because not having enough surface area for it to happen, basically just running out due to the overwhelming granuloma formation in the lungs. I think they say Doc Holliday, they believe, died of tuberculosis potentially, right? Some of the movies, I think they might even say it.
I remember in the old books, they used to say, and they died from consumption.
And I was just going to say that. And a lot of times it was referred to as consumption, right? Tuberculosis was referred to as consumption. The really scary thing about that is highly contagious. As little as 10 bacteria, you could potentially inhale from someone else and get the infection. Yeah. And so that's another thing, like when I talk about people and we talk about what are called face talkers, like people that talk right in your face. Don't back up because they'll just keep coming at you, right? You put your hands up and you create that barrier, right? Like, do not talk in my face. This is my zone. This is my protective zone. And there was one commercial they had on TV for the longest time. I would just giggle when they had it on. They said, "Don't huddle up with sick people." In other words, don't get really close to them, right? Keep your distance, because some things are spread respiratorily.
I don't know how, because between the military and then working at Charity in Lafayette, and we were ER, and we had TB patients come in, and they were in crisis, but I don't know how I never managed to become positive to having been exposed.
Yeah.
When you do your TB times or...
You were careful, hopefully.
Oh, I'm still a negative.
Yeah.
But I mean, I remember when we were teaching them in the military, it's not a big deal that you give a positive. We're going to do a chest x-ray, make sure you're not sick.
Right, yeah.
And after that, you got a year and a half of medicine.
Right, yeah.
And then you're a converter.
Yeah.
No, can still come
Yeah. Once you're positive for the TB test, you'll always be positive. And we'll talk about that as we talk about how your immune system responds to pathogens and the memory component that we have for our adaptive immune system. And we'll specifically talk about the TB test when we get to the later, and how some people don't actually have the infection, but from being exposed even to the TB test numerous times, you could essentially be vaccinated by the TB test, and therefore you'll always be positive. So that runs into some nurses that, some of you guys, this may happen to you later on in your careers. Don't panic, right? It doesn't necessarily mean you have tuberculosis. It just means that you've been exposed to the level where your immune system now remembers it. And once it remembers it, it will always remember it. And so the same thing with vaccination is we're exploiting that memory component, and we're exposing you to it so that you remember it and you respond faster the next time you're exposed to it. So some countries, they do vaccinate for tuberculosis. So sometimes two people come into this country and they get TB tested and they come out positive, and that's the reason, is that they've been vaccinated, and they'll always be positive. So do you guys understand this component? This is paramount to our immunological response, right? Getting these white blood cells to the areas that they're needed to do their job. So, damaging effects of inflammation. The problem is that this is kind of like a sprinkler system, right? Once you set off this system, right, you've got those suicide bomber neutrophils. I mean, they're just going nuts, right? And depending on where this happens, this can be damaging to the body. But the whole idea is fast acting, right? Keep it in that particular area. So a lot of times it's kind of equated to a sprinkler system. Sprinkler system is great if you don't want to lose the building. But if you're talking about a computer lab, you just lost all your computers, right? If the sprinkler system goes off. Electronics and that doesn't quite mix all that well, right? So there are enzymes that are toxic that are released by these phagocytic cells. So sometimes we're even damaging our own tissue while we're trying to kill the invaders, right? It's not a very specific attack. It is specific to an area, but we don't necessarily discriminate between our cells that get in the way. Make sense? Okay. So in our finger, not such a bad deal, right? We can minimize it, we can localize it to a particular area. Delicate systems, like the meninges, which is the name of the membranes that surround your brain and spinal cord, those are really important areas of the body, right? Kind of control everything, just a little bit, right? And because they're so important, they're encased in what? Bone, right? And is bone flexible when you're an adult? No, right? Those things have sealed up. That skull is solid, right? And it's meant to protect that very important organ. Same thing with your spinal cord, right? It's protected by your vertebrae, although not quite as encased as the brain, but pretty well encased. There's no room there for flexibility, unlike with their skin, right? Where we can swell, right, and swell out. When you swell with the brain, right, it's pushing back on it, right? In the lungs, inflammation in the lungs decreases surface area for breathing, right? And so therefore can really interfere with the ability to breathe. So those are the two main areas where inflammation, if prolonged, right, and doesn't quickly take care of the problem, can actually be life-threatening, right, and even deadly. That's why they freak out about these things, right? Pneumonia, it just means an inflammation in the lungs, right? She's like, yeah, don't even get me started on pneumonia. Right? And there's lots of different things that can cause that infection. A virus can, bacteria, fungus can cause infections in the lungs, lots of things. Pneumonia just means an infection of your lungs, right?
Those are nasty ones.
Yeah, and again, because you really need that ability to breathe, and having inflammation there really impedes that ability, right? So cell death in the inflammatory process. So when our cells die, sometimes they're programmed to die. They've finished their job, their lifespan is done, it's over. And so they'll be either signaled themselves or signaled by other cells that it's time for you to go to the afterlife, right? This is referred to as apoptosis, right? Some people refer to it as cell suicide. This does not trigger the inflammatory process, because think about this. Your cells are dying all the time, y'all. Red blood cells only make it 120 some odd days, right, before they're just spent because of how much work they do, how much traveling they do. They're long distance truckers, baby, right? You can only do that for so long before you're like, I'm done, right? It's a tough job, okay? So for them, right, they'll be broken down, right? We even recycle some of their components, right, especially that good old iron. When they die, if we had inflammation, we'd be in a state of inflammation all the time. all the time, probably everywhere in our body, if we were in inflammation every time a cell died in our body, right? So we have to have a way for them to go away, right, and new ones to come online without having that inflammatory response. Now, pyrophytesis is if patterns are recognized, right? If there are triggers, right? If receptors on our cells recognize danger, right? Then we are going to elicit inflammation, right? So we have control over this. It isn't just cell death, right, in general, right? It's cell death under certain conditions is going to elicit inflammation, right? And that's the important distinction there. All right, so the next thing that we're going to look at is phagocytosis, right? So when the phagocytic cells get their specialized white blood cells, that this is their main job. Our two main ones are macrophages and neutrophils, as you guys saw in those pictures that we've looked at already, right? We talked about these guys. But there's some similarities and differences, important notes about these guys that we'll address. But first, let's watch them in action. So I have my real videos of the real thing.
There's the neutrophils.
Yeah, so here's our neutrophil chase.
Neutrophils are white blood cells that hunt and kill bacteria. In this spread, a neutrophil is seen in the midst of red blood cells. Staphylococcus aureus bacteria have been added. The small clump of bacteria release a chemoattractant that is sensed by the neutrophil. The neutrophil becomes polarized and starts chasing the bacteria. The bacteria, bounced around by thermal energy, move in a random path, seeming to avoid their predator. Eventually, the neutrophil catches up with the bacteria and engulfs them by phagocytosis.
They got the cute little sound effects and everything for this one. So they literally just took blood and put it on a microscope slide, added some bacteria, and filmed it. Now, I'm not sure if they've sped this up. It may be sped up. I'm not sure. They don't indicate. So what you're seeing here is they said are red blood cells. Here's that neutrophil. It almost moves like an amoeba. Remember, there is liquid in your blood. Your blood is mostly liquid. So that particular bacteria, as they said, is non-modal. And the only reason it's moving is it's in a fluid matrix, so it's kind of being bounced around by molecules colliding with each other. That's what they mean by the thermal energy. Does that make sense to you guys? But again, he's releasing stuff. He basically smells bad to us, right? And we're chasing after him, right? That neutrophil can sense him, and he's going to get him, right? And he does. And so, here, I got to shrink. So the other one, again, really shows you how specific that interaction, that kind of smelling is.
Taken from the blood of a graduate student are mobile cells that will quickly migrate to sites of injury to help fight infection. They are attracted there by chemical signals that are released by other cells of the immune system or by invading microbes. In this experiment, tiny amounts of chemoattractant are released from a micropipette. When neutrophils sense these compounds, they polarize and move towards the source.
So they look like an amoeba, huh?
Yeah, kind of. Smells good over there.
When the source of the chemoattractant is moved, the neutrophil immediately sends out a new protrusion, and its cell body reorients towards the new location.
Yeah, they had fun with the sound effects. Oh, they're cute. And my computer says if I don't plug her in, she's failing on me. So we better make her happy.
I don't like that program itself, but only little so
Okay. All right, so that's it for these. I'm gonna close these down. attempt to close them down. I don't think there was anything more I wanted to say about those. Let's click the right thing, Erica. So one more, then I promise we're done with videos and stuff. But it really helps, I think, right, to see these things moving instead of just talking about it. Okay. So this one is going to go into, again, a little bit more detail.
In phagocytosis, phagocytes are attracted to the area of invasion by chemical products of the microorganism, phospholipids released by injured mammalian cells, or by components of the complement system. The phagocyte moves into the area of invasion and then attaches to the microorganism. The attachment is mediated by a variety of surface receptors, including antibody, lipopolysaccharide, and complement receptors. For example, the C3b component of complement coats bacteria or other particles and then binds to C3b receptors on the phagocyte. This process of coating to enhance phagocytosis is called opsonization. The microorganism is then engulfed by the phagocyte into a vacuole known as a phagosome. Vesicles in the cytoplasm called lysosomes fuse with the phagosome, releasing digestive enzymes such as lysozyme and proteases into the phagosome. The structure resulting from this fusion is called a phagolysosome. Inside the phagolysosome, microorganisms are killed and digested. Finally, the digested contents of the phagolysosome are eliminated from the phagocyte by exocytosis.
So we have several white blood cells that have this ability to do the phagocytosis, where they bring it in, and as I said in the beginning, right, they are able to sense, and as you saw, they have receptors on their surface that even facilitate that binding, right, being able to grab on to, say, that bacterial cell and bring it in. You had a question?
I thought phagocytosis also presented.
Right. So one of the things that they're not showing you here is some cells, some phagocytic cells, go one step further after they've digested it. They're going to take pieces and present it on the surface. That's called antigen presentation, right?
So the body knows what it is.
Yeah, so the body knows what it is and can recognize it. Yep. We're just focusing on the killing part right now.
Does it only do that with things that are new that have never been seen before?
No, it does it with everything. It engulfs, right? So, but not all cells can do antigen presentation. We'll talk about the ones that can. Not all phagocytic cells. So for instance, the neutrophils don't do antigen presentation.
So the complement that is coating the bacteria is from the phagocyte?
So the complement, this C3b protein that they talked about, is part of a system we have in our body called the complement system. It's a series of proteins that we're going to go through and talk about later. And these proteins, this specific one, I always remember C3b for binding, can bind to the surface of bacterial cells. And when it does that, it aids in our phagocytic cells being able to grab onto it and engulf it and destroy it by phagocytosis. And then they also refer to it as an opsonin, right? Let me pull it up so that you can see that word. I know they did. There, opsonization, right? When you coat something for destruction, so C3b is an opsonin because it will coat them and phagocytic cells will grab on and destroy it. Another opsonin we'll talk about are antibodies. So again, these are products of our body, of our immune system, that attach to things like bacteria or viruses and tag them for destruction by these phagocytic cells. But there are other things that they can recognize, right? They have other receptors. So notice that they said things like lipopolysaccharide. And you guys remember gram-positive or gram-negative bacteria have that. Lipopolysaccharide. It's part of the outer membrane. Gram-negative. Gram-negative have that. And so these are general characteristics about bacteria. That's not very specific to E. coli per se, right? E. coli has lipopolysaccharide because it's gram-negative, right? But our system isn't necessarily targeting it because it's E. coli, it's targeting it because it knows it's a bacteria, right? And that we know that it therefore could be dangerous. Make sense? Right? So we have these general patterns that we can recognize, and then we have things that are very specific we can recognize, and that's where antibodies come into play, right? And other cell receptors that we'll talk about in our adaptive immune response, right? So we have different layers to this response, right? And how how it orchestrates. So, as I said here, chemotaxis, right? In this case, this is an example of another complement protein, C5a, and this one is a very strong chemoattractant. It is a cytokine, it's a chemical message, right? Cytokines are chemical messages that are going to draw the white blood cells to that area. And in this case, too, C3b has bind to the surface that helps facilitate the phagocytes attachment, right? They got to be able to grab onto it. And your phagocytic cells have lots of different types of receptors that allow them to grab on lots of the different types of danger molecules, right? Patterns that we see, lipopolysaccharide, peptidoglycan with the cell wall of gram-positives, flagellin, that protein that makes up the flagella.
For the picture we're using on our test, will we have to know kind of the process of how?
Yeah, we need to know the steps of the process of what's happening.
Do we need to know like the different C5a, C3b? or
You will eventually, yes, need to know C5a and C3b.
Thank you.
Yep. All of this whole slide is all pertinent information. There isn't anything in here that we're not going to talk about. Okay, so the name that we give to the vesicle when it first engulfs the pathogen is referred to as a phagosome. When that phagosome fuses with lysosomes, we refer to that as a phagolysosome. We basically just fused the two names, did we not? And that's the same thing that happened with those two vesicles. And so with these vesicles, they're releasing digestive enzymes that are in the lysosome, right, that are going to gobble up and destroy the bacteria. And they're going to kick out the extra stuff. And one thing that's shown here that some of your phagocytic cells will do is they'll pick up pieces of this pathogen and they're going to present it in receptors on their surface and communicate with other cells of our immune system. Right? That's a whole different step. We refer to that as antigen presentation. And we're going to get into that in our adaptive immune response because that's where the cells, the white blood cells, communicate with each other via antigen presentation. So the other thing here is we still get sick, don't we? And I already talked about one bad guy, right? And I think it is that even when the lysosome fuses, Mycobacteria tuberculosis can grow inside that phagosome. Those digestive enzymes don't destroy it. And then there are other pathogens like Shigella. They can escape the phagosome. They get in and they just jump right out. They're like, yeah, thanks for the free ride. I'm going to go someplace else right now. Right? So although we have these defense mechanisms, right, there are ways for them to get around it. There are ones that can stop C3b from binding. So that makes it harder for our phagocytic cells to find them. And then some of them we've learned about how they have a capsule. Some of their capsules help protect them, make it hard for these phagocytic cells to attach to them, recognize them, and engulf them, right? Shigella, too, is another one where it can use our actin filaments inside of our cells and shoot from one cell to another, like slingshot itself. So if it's traveling through our cells, that makes it harder for us to get to it, right? It's like running behind the lines. Yeah, catch me if you can. You know, so what we refer to those as virulence factors, right? What is it they can do that evades our immune system or makes them toxic for us? Lots of them produce toxins too, right? That damage our cells and damage our body, right? Some of them, like I said, just, evade phagocytosis long enough to cause even more damage and reside in our bodies. So for each one of these things that we look at, defenses, there's a bad guy out there that's found a way around, right? Just like, if you have an alarm for your house, doesn't guarantee you're not going to get broken into, right? It just means you made it a little bit harder and you're going to get the really smart ones that break in, right? As opposed to the passerby.
You're going to have a better class of criminal.
Yeah. I don't know if that's good or bad. Right. So your two main phagocytic cells are your macrophages and your neutrophils. All right. So these are specific groups of white blood cells. Your macrophages are always in your tissues, right? So that was, remember that was that resident one that was sitting there, right? Always chilling, kind of always on guard. If something gets in, it's going to attack. And it's going to call more of its friends to the area.
So the monocytes, while it's in the blood, does it
It
still
can
have
do-
an immune response, or
Yes.
it only
Yeah.
once it's in the
Yeah, so the name for macrophages when they're in your blood is called a monocyte. And it can still do the same stuff in your blood that it does in your tissues.
Are you going to use the term fixed macrophage or resident macrophage?
Probably not necessarily neither. I would say macrophage is found in your tissues.
That's better.
Yeah, more specific. I wouldn't necessarily use the resident or fixed macrophage term. Right? I would say the ones in your tissues. And in fact, when they were first discovering these cells, and we'll talk about this again later, macrophages that are in your liver were referred to as Kupffer cells, right? Because of course, probably someone named Kupffer discovered them, right? And then come to find out later, we're like, oh, well, it's just a macrophage, just happens to be in your liver, right? So the good news, I guess, by the time you got to your lungs, they just called them alveolar macrophages, right? And that's just to say that it lives in your lung, right? That's its home, it stays there, right? It's on guard.
Do the ones that kind of live in certain areas have the receptors to notice?
They all have receptors to notice when bad guys have gotten
But
it.
I mean, do they specify, like, is the ones in the lungs more ready to catch the things that come into the lungs than, say, come into the
Ooh,
kidneys?
good question. So her question was, are the ones in your lungs more geared for the stuff that come into your lungs as opposed to the ones in your kidneys are geared up for the particular stuff that may come in contact with your kidneys? I would say probably so, but I have no fact to back it up. I would say it would make sense, right? Because the other thing too is that the type of lysosomes and stuff that they have, that can be changed. And I would think that that would be environmental too. What do we most experience here in this area? What type of toxic substances do we need? Do we need peroxidase? Do we need other hydrolytic enzymes?
That makes sense. That's just
Yeah.
like allergy testing. If you're in Illinois, you're going to test for certain things, but if you're in Louisiana,
Mhm.
you're going to test for
Environment
certain things.
makes a big play on all our cells in general. Okay, I'm good on time. Okay, these guys survive after phagocytosis. So one of the things that we had mentioned, things like antigen presentation, these guys can do that because they're going to engulf it and be still alive, right, and be able to present it. Where, especially if the neutrophils degranulate, if they release their granules into the area, they die, right? So, and like I said, they're very short-lived cells. These are the really powerful killers, though. Their lysosomes and stuff have really toxic substances in them. And these guys have, as I said, the greatest killing power, and they can even shoot their cytoskeleton out and create nets to try and trap the pathogen. So this is one of the newer things that I learned even after I took immunology, was their ability to shoot their cytoskeleton out of themselves and create these nets, right? So they're basically, like I said, they're killing themselves in an effort to trap the stuff in that particular area.
You had mentioned earlier that, like,
Hold on, guys, he's got a
pus
question.
Is dead neutrophils or something like
Yeah.
that? Isn't usually pus a sign of infection?
Yeah, pus is definitely a sign of infection because you're having lots of white blood cells dying. And what's the reason for that? Well, they've been called to that area probably because it's infected, right?
About the pus, since he's mentioning it, what causes the different color variations of it? Is it because of the infection or what it's fighting? Like how it can get really green if you're really infected or yellow?
Yeah, the different colors have to do with the different components, and I don't know the exact components to what colors. But yeah, it has to do with what's going on there, like what pathogen is there, what cells are
You haven't got
there.
any good videos on pus?
I probably could find some good videos on pus on the internet, but I was pretty sure that'd be a little too much for class. But you guys can look that up for yourselves. I've seen some stuff that I don't even want to watch. So I know they're out there, but I'm not going to subject you guys to that in class. It's bad enough talking about it sometimes. You don't really need to see it. Sometimes we've seen this stuff.
Just think of a pimple. It's the same
Yeah,
concept on
A
micro
pimple,
scale.
a pimple is, usually full of pus. It's an infection of a hair follicle in your skin, right? Which is also why you don't really want to pop them, because then you're spreading bacteria to other parts, right? That's why you want to clean it up. Little triple antibiotic ointment, y'all. Works wonders. Yeah.
Tell her about
Bacterial infection.
that. She's having a rough time.
Yeah. So again, I mentioned earlier, giant cells. This is when the macrophages will fuse together when we're usually in a chronic type of inflammation situation. And when T cells become into it, it's a granuloma formation. And again, it's just piling up of their cells. They're trying to wall it off. They're trying to keep it in a particular area. They're not being successful in killing it. Now, as I said, macrophages can communicate with other cells, so they can communicate to T cells. And this is when they're doing the antigen presentation. And this is when the T cells can help communicate with them to change what killing strategy they're doing and make them be more powerful, right? So they have a bunch of different, you could say, missiles in their arsenal, and the T cell kind of helps them pick which one they should try next, right? Or what's been proven against this particular pathogen. Because this is going to be a specific interaction. These T cells are going to recognize that actual pathogen that the macrophage is presenting, okay? And there's one other cell that does phagocytosis, but really is the specialist with antigen presentation, and that's the dendritic cells that we'll talk about later. So they're still going to do this process of phagocytosis, but they're really the specialists that go and share this information with the other cells, like our T cells of our immune system, and really amp up our ability to respond, right? So here are all the groupings of cells, and next time we'll go into more detail. I'm just going to introduce them. And some of them are going to even go into their functionality a little bit more. So we're really going to focus on that functionality, but I'm going to give you guys some tips that are in A&P on how to identify these cells and know the different groupings. So under the grouping of granulocytes, we have neutrophils, eosinophils, and basophils. And one of the things that all these guys have in common is the granules, hence why they're called granulocytes. And so when scientists were studying these cells and they stained them, they noticed that they stain differently for each one of these cell types, right? So these are the little circles that you see inside. These are the granules. And you'll notice, and I don't know if this is on purpose or not, but all their names end in "phil." So I always remember all the phils are filled with granules, right? And then the first part of their name actually has to do with how their granules stain, right? So when you do the special staining procedure to be able to differentiate the different white blood cells, neutrophils, their granules stain relatively neutral. Sometimes they kind of blend in with the cytoplasm. You kind of don't really see them. Eosinophils, their granules get dyed with the dye called eosin that's in this dye mix. And so then they come out a reddish to orangish in color because of that dye. Basophils are basic staining, and they're blue or purple. All the nucleuses for these guys stain purple as well. But it's not the nice round nucleus you're used to seeing, right? They're what we call multi-lobed. So they're stretched out, right, in these different lobes. So when you're trying to identify them in A&P under the microscope, the first thing you should do is look at the nucleus. If it's multi-lobed and you don't really see any granules, it's probably a neutrophil. That, and they're the most abundant white blood cells, right? And they're actually even going to increase even more with inflammation, with infection, because what's their main job? Phagocytosis, right? And digest and kill, right?
The body, in fighting infection, will produce more
of them.
Three,
Yeah.
because certain levels you can tell whether it's a bacterial or a viral or a parasitic infection just by the white
Yep.
blood cells.
Yep. So what Carol was referring to as is that the reason why we have this staining procedure where we could look under the microscope, and this test is referred to as a differential. And what somebody like myself would do is we would do a smear, of someone's blood, and we would stain it, and we would look under the microscope using a special counter, and we would identify each one of the major 5 white blood cells that you see in your blood until you get to 100, and usually the special little thing dings, right, indicate that you've reached 100. So percentage-wise, you can see the percentage. Now, normal percentage, you're going to have a lot of neutrophils, but under infection, you're going to have even more neutrophils, right? And different types of infection, you're going to have different increases, right? Different variances in these percentages from normal. So for instance, for the next one, for eosinophils, notice these are important in the inflammatory response, again, because what their granules are. But for some reason, too, these increase when we have parasitic worm infections. They go through the roof. And so, again, they would be able to see, if they looked at a differential and you saw this really big increase in eosinophils, they may look, hey, we may have parasitic worm infection, right? Basophils, their multilobe nucleus sometimes is obscured by their granules, right? So it just looks like this big purple mass, purplish blue mass. And so a lot of students in A&P confuse these with, it's depending on what magnification you're looking at, with lymphocytes. Lymphocytes have a nice big circular purple nucleus with this staining procedure, and sometimes just the tiniest little light blue cytoplasm, right? And if you're looking at those guys at high power, their nucleus can sometimes look a little granular. And if you're not looking very carefully and you miss the light blue cytoplasm, you might think you're looking at a basophil. Trust me, you probably are not, because they're extremely rare, right, in blood. Although I argued once with my immunology teacher when I was in grad school that I was pretty sure because we were able to take our own blood then. Back then you could mess with your own blood in lab. And we did differential stain, and I could have sworn I was seeing basophils. And she's like, no, there's no way you're seeing basophils. I'm like, I can swear that's a basophil. And you know, it probably was, because I have horrible allergies. Right? And probably at that time I might even have been experiencing allergy. And so these guys are the ones that are packed with histamine. And when they're in your tissues, we refer to them as mast cells, but these are very close cousins to the basophil, right? And that they contain histamine. The only difference is the mast cells really are resident in your tissues, where the basophils are traveling through your blood. And again, they're going to be called up like maybe when you're experiencing allergy, right? You may produce more of them, and therefore you'd see more in your blood.
With the lymphocytes, is there some kind of marker that we differentiate between T lymphocytes, B lymphocytes, or is it just by what they're doing is how they're saying, okay, this is going to be a T lymphocyte? How do they know the difference between them?
There are markers.
There are markers,
Yeah.
like protein markers.
Yeah, let me get to that one. You're jumping ahead on me. It's okay. It's okay. It's an excellent question. It's an excellent question, and I will get to it. I just want to stay in order so I don't forget. Okay. So then our mononuclear phagocytes, and again, these guys are really named after their nucleus really kind of stays intact, right? Although it tends to have a little indentation in it sometimes. And as I said, monocytes is the name we give to the macrophages basically when they're traveling through your blood. And they have the same abilities, right? They can phagocytose materials in your blood, okay? Your macrophages are in what we call in your tissues. And when your body makes these, they travel and they inundate all throughout your body, all your major organs. You have resident ones that live in those tissues, right? And ones all along your skin, I mean, just everywhere. Right? They're everywhere. They're on guard everywhere. And their main job is phagocytosis. They can do antigen presentation, but their main job is phagocytosis and killing, right? Walling them off, keeping in that area, killing them off, okay? Dendritic cells, on the other hand, which are believed to be descendants as well of monocytes, these guys are named after, you notice that they're drawing these long tendrils. These guys, again, are throughout your body as well, but they travel. They're kind of like the scouts, okay? Once they've encountered something And these guys can literally grab those tentacles and go in between endothelial cells and like reach into your gut and grab stuff and sample it and say, let me see what this is, right? And like if it's something dangerous, they'll take even more, right? And then they run off to areas where you have large collections of your lymphocytes that we'll talk about next and presents that information. So their main job, right, is collecting data, right, gathering it and giving it back to the lymphocytes. And most of your lymphocytes are collected in particular areas in your body, lymphoid tissue and lymphoid organs, right? And they're waiting for their dream antigen, right? Because they're antigen specific when we talk about B cells and T cells. So one of the things that he was mentioning earlier, right here we just say they're involved in the adaptive immune response. And for that standard counting the white blood cell thing that we do from your blood, you cannot differentiate the different types of lymphocytes. And there are actually 3 different types. 2 of them that are antigen specific. They're commonly referred to as the B cells for short, or B lymphocytes, and your T lymphocytes. And then the other one, 1973 named, so it's the awesome name, right? And it's the best year because I was when I was born. Natural killer cells, right? You won't ever forget that, right? Natural killer cells. If 1973 named them all, we'd have all kinds of interesting names. So underneath the microscope, if you're just doing a standard differential with the standard dyes that we use, you're not going to tell B cells from T cells or natural killer cells, right? You're just going to term them all lymphocytes. They're all pretty much going to have a pretty nice round, for the most part, nucleus, little tiny light blue cytoplasm. Now they're showing you 2 different colors here just to denote that there are 2 main types of lymphocytes that we discuss. Now he asked, well, can you tell the difference between them? Is there markers that you can tell the difference? And there is. And so they have actually developed stains and different machines and techniques that can actually separate out the 2 major types of B cells and T cells. And it's because they do have different markers or receptors on their surface. You guys want me to clue you in on those now? And then we're going to talk about it again later? Okay. All right. So this is really important, too, with another disease. I'll help you remember this, is HIV infection. So HIV specifically goes against T cells, but even within the T cells, a particular population of T cells, right? So when we talk about T cells, we're going to talk about helper T cells and cytotoxic T cells. And a lot of scientists nowadays just refer to them by their distinct markers. So CD4 helper T cells, right? CD4 is clusters of differentiation. It's a marker on their surface that they use to communicate with specific cell sets that helps us determine them. And unfortunately, the CD4 helper T cells that kind of help out a lot of our cells are the ones, that CD marker is what the HIV virus is using to attach to our cells and get into those cells. Now the problem is those are our immune cells, right? They help out with our immune response. If they're infected with a virus, they can't help, and they're going to die. And so then you get to a point where your immune system becomes what we call deficient, and that's AIDS, acquired immunodeficiency syndrome. These 2 are not synonymous. Not nowadays. There are people living with HIV infection that are not progressing to AIDS. Why? Because we have treatment now, right? We have some antivirals and such like that they stay HIV infected, right? Sometimes they're thinking they've even cured some individuals. That would be great if they've eliminated the virus from the body. Not an easy task, but it'd be great if we can get to that level. But keeping it at a level, and this is why a lot of times they'll do blood tests, and they're specifically very specialized tests to see where the levels are for the helper T cells. How debilitated is that population by that virus infection? So yes, we have a way of distinguishing B cells from T cells, even T cells from each other. And so the other T cells that will hunt and kill virus-infected cells, like unfortunately your helper T cells in the case of HIV infection, is the cytotoxic T cells, and their marker is CD8. And so how I remember this is 8 kind of sounds like eat, and cytotoxic T cells basically eat or kill other cells. And then for CD4, I always remember 4H, right? 4H, right? For CD4, H for helper T cells, right? Or, the organization 4-H that helps kids, right, learn about animals, right? You got to tell yourself little stories sometimes with all these numbers and letters and acronyms, right? And so I love immunology, but I'll tell you, the number of acronyms is mind-blowing, and you guys are only going to experience a few of them. But I'll teach you my tips and tricks as it comes to that, right? So we're going to go over that and look at that really specific interaction later. And as I said, we're going to continue to discuss the roles these cells play, right, and where they come from. And I will, right after class, go to my office, post the next PowerPoint for Tuesday. Work on your case studies. Hold on.